And I have a lot of function, which is nice. Right. Something like this. Yes. Hi. Yube.
Sorry, it's a mistouch.
No problem. Problem. I don't know what to do with this. You don't see me very much, but never mind. You don't need to see. You know what I look like. Okay. So now I'm going to start, and let me share my screen. Oh, it's this guy. Right. So this is a short lecture about point sprite function and resolution. We have already spoken a little bit about that before, at least how the point sprite function forms. So I remind you that the light that is emitted by the sample, some of it makes its way into the objective, and at the edge of the objective, there is diffraction that happens. And then the objective, because of that, the objective edge acts like an aperture. So it's an iris outside of the image plane, and that means it affects the whole image. It doesn't work like a curtain, it affects the whole image. So this is what I've shown you before. Then at the end of the whole optical train, we end up having the final image of our sample, and this is a 3D interference pattern, and this is what we actually record. So this is what the image, so a little fluorophore, which is a tiny sphere, which has the same dimensions in all directions. In the XY direction, it looks like much bigger blob with some rings around. And in the Z dimension, if you make a Z stack through this, we see a rugby man. We see an elongated blob. So we have a different resolution in XY and in Z. And the image of every single fluorophore in our sample does that. And this is what we're imaging. We're imaging the point spread function. So this is the rugby man. And then I played a little bit with AI and I thought, yeah, what happens if you have a lot of fluorophores in your sample? And this is what happened. You get like a scrum of rugby mans with lots of rings around. And this is what AI gave me when I say, give me some rugby man with rings. So this ring is not really finished. But anyway, that was a while ago already that I did that. So what happens when we have many dots next to each other? How did the point spread function relate to the resolution? So here we can see real images of small beads. Well, I think I'm not sure if it's real images or if it's simulated, let's say. So we have these two point spread functions that would correspond in our sample to two small dots that are fluorescent. And here they're quite far away from each other. By the way, these images come from also the same eye biology microscopy course by Jeff Lichtman, and it's on YouTube. So when the two small spots in our sample are far away from each other, we have the two point spread functions do not interfere, where the rings interfere, but not the main lobes. And then as the spots come closer to each other in our sample, the two main lobes are coming closer to each other. And if we have if the two point spread function main lobes are one point spread function diameter, so the main lobe of the diameter of the main lobe, if they have that distance between them, we can still resolve them. Okay, now we have one diameter of the main lobe distance between them. But if as we get closer and closer, if we go to one radius, so if the distance between the two point spread function main lobe is one radius of the point spread function, then we cannot distinguish anymore, and then we end up having just one blob. Basically, we cannot tell if it's one or two. So the resolution is the smallest distance between 2 points in the sample that can be reliably distinguished. Clearly, it depends on how you analyze. And therefore, as I said, a small distance is actually a high resolution. And one thing that is important to remember, if any object is smaller than the point spread function, it will be represented by the point spread function. So it doesn't matter what size we have, it will look like, so even if this object is super tiny, what we are imaging is the point spread function. So as long as the object is smaller than the point spread function, it will be represented by the same point spread function, okay? This is the minimum, the macroscope, the objective that we have with that wavelength. This is the minimum it can represent, even if the object is much smaller. Okay, so objects that are smaller than the point spread function anyway look like they have all the same size, the size of the point spread function. So therefore, the smallest distance that we can resolve between two dots is the radius of the point spread function. So now I'm going and justifying myself. Why did I pick these two equations for the course? There are so many definitions of resolution online. It is completely, I tried for many years to try to make sense out of the whole thing. At the end of the day, what I think is because for many of the equations that we find online, there is no context, and that's the problem. So we have here a radius. So the two blobs are one radius, more than one radius away. We can distinguish them when we become one radius. We can just about distinguish them if they become closer to each other by one radius. Then we cannot distinguish them anymore. What is this radius? So this is again like an intensity profile through the XY image of the point spread function. And what you can see here is that the radius is going to be this distance in here. And this actually corresponds to the Rayleigh XY optical resolution 0.61 lambda divided by NA. So this is the equation that we will use, and that corresponds to the radius. And this is what it means in real life. But this equation is actually empirical. This guy was quite a long time ago, I think in the 1800, like early 1800 or late 1700. And what he was doing is like, he was looking with his eyes and going like, now I see two, now I see one. So it's completely empirical. But that equation somehow stuck throughout the ages. Now we have other equations that can maybe represent better the resolution of a macroscope that we do not have to look with our eyes. One of them that is used a lot is the full width half max. So what is the full width half max? This is the full width. So this is the maximum. So here is the maximum of the main lobe, and here is half. And if we look here at the width of the half, this is what the full width half max is. So you can see that it is a little bit different. It's a little bit smaller than this one, because this is 0.51 and this one is 0.61, okay? So it's not super much smaller, but it's more optimistic because it's smaller and the small distance means a high resolution. So we like that. Anyway, full width half max is used a lot to represent the resolution. And then there is the Abbe limit. And the Abbe limit, instead of being 0.51, is 0.5, so it's half. Okay, that's the one that's engraved in stone in Jena. But that one, as I understand it, again, I'm not a physicist, as I understand it, this is actually a hard limit. And this is theoretical in the sense that at the Abbe limit, you have no contrast. And if you have no contrast, you cannot use the resolution anyway. So, okay. And then there are some equations that use the excitation, some of them that use the emission wavelengths, and that depends on what you do. Normally, for a wide field system, we should use the emission wavelengths, and for a confocal system, we should use the excitation wavelengths. So that is the normal way of doing it. And when we come to Z, so the depth of field is this resolution for wide field. That's the equation that we use, that we will use during the course. And this is for wide field. And it represents also the radius, but in the Z dimension. If you use a confocal and your pinhole is open over one area unit, the equation becomes this, right? And you can see it's lambda excitation. I said no. I said in this course, we're going to use two equations and we're going to use them for everything. And I understand that it's not very scientific, but at the same time, what I want is that I want to make it easy for you to understand the principles. And then after the course, you can use whichever you want. Just take with a pinch of salt what you find online. There's a lot of mistakes on top of being lots of things without context. So in this course, we will use the radius of the point sprite function for x, y, and z for all systems. And we will use the emission wavelength. And so this is going to be 0.61 emission lambda divided by numerical aperture for x1 and 2 emission lambda n, so that's the refractive index of the immersion medium, divided by n squared. That is the way we're going to be doing it. And just we will consider that if you close the pinhole, then you are getting better contrast, and therefore you can use the resolution that the objective is sending you. So, and then if one area unit, as we have said, is a way to normalize the size of the pinhole, and this is basically the diameter of the point sprite function multiplied by the magnification. So we're going to use this equation with the emission here, and then multiply by 2 and magnification. This is what we're going to use, and that's it. So anybody shoot me, it's okay. So I've done my little explanation of why we use these resolutions now. And now I want to show how the resolution and the sample design are connected to each other. So if you count some small dots like this that are crowded, you need a high resolution that is needed. But if your dots are far away from each other, they have the same size, but they're far away from each other, so they're sparse, then you can get away with a low resolution. You anyway need the high sensitivity because you want to detect the dots that are small, so presumably they're not super bright. But so you want a higher sensitivity, but you can have a low resolution because they are far away from each other. If now we're looking at big objects, if they are crowded like this and you have labeled them in this way, for example, nuclei in a compressed area in a tissue, very often they are elongated and they're flat, they're very close to each other. So this type of objects, if you label them with DAPI, for example, this is what they look like, and there's very little distance between them. If you label your DNA this way, you will need a high resolution because what you want to resolve is actually the distance between them. In the same way as here, if we have a small distance between them, we need a high resolution. We have a long distance between them, we're fine with a low resolution. It's going to be the same thing here. Now, if your sample is like this, you have other options. For example, you could label, if it's a DNA, you could decide to label the nuclear membrane instead. And if you label the nuclear membrane, your image will look like this instead of looking like that. And that makes a very big difference for the way you analyze the images, because in this case, you could easily differentiate between one border of the nucleus to another. And this is now what becomes limiting for the analysis. So you can see how dense the signal in your sample is plays a very big role in how you're going to analyze, and therefore what type of resolution you need, and therefore what type of objective you need. And it does not depend so much on the size of the objects, but more the distance between the object, and that talks to how you label. So you have to think about all these sort of aspects when you prepare your sample. Are you going to make it difficult for yourself to analyze or not? If you have objects that are very crowded, you want to try and find strategies to identify them in a way that makes it easier and more robust. So the design of the sample influences the resolution required to answer the scientific question. And the required resolution is not so much related to the size of the object, it is more related to the distance between the objects. Now, again, if you have one object, even if it's very small, you can detect it without a high resolution. So you need to reverse think your experiments according to your knowledge of the sample's biology. So if you know that you can label this way instead of that way, this will help you identify how many nuclei are in a crowded area. So the conclusion for this short little talk is that the resolution is the smallest distance that we can reliably measure. And it is not possible to resolve distances smaller than the point spread function radius without super resolution. So we can with super resolution, but not without. Okay. So any object that is smaller than the point spread function will look at this, like the point spread function. Exactly. I'm ahead of myself. In this course, we will use for camera-based and single-point confocal microscope this equation for the optical resolution, and this is the emission lambda. And I forgot to write it down. And then we will use also for the Z resolution this equation. It's also the emission lambda. We will use this for everything. And we consider that if you close the pinhole, you will increase the contrast and the usable resolution. And we need to realize that if you close the pinhole more than one area unit, you actually increase the resolution. And this is something that if you have a very bright sample and the need for high resolution, you can think about. You do not have to put your pinhole at one area unit systematically. You can open it bigger if you need more photon, and you can close it a little bit smaller if you have very bright sample and need the extra resolution. Okay, but when we're above one area unit, closing the pinhole is actually increasing the contrast. So we can use all the resolution that the objective is sending us. Okay, and now we're going to go to, I look here at my stuff. Yeah, now we're going to go into the workshop Zoom, and we will do some exercises. So everybody, people online, if you're interested in the next lecture is at 1, and everybody else, I see you in the workshop Zoom.